event.
Hello, everyone. Thank you for joining this webinar. Thank you for your time. And I would like to, first of all, I would like to promise that I will make it as short as possible, because I know you're all busy, you're doing your experiments, you are swamped with many other things. So, and I'll try to squeeze it into 20 minutes so we have enough time to for answers and for questions and answers. Yes, as Francis introduced me, I'm a scientist with Bio-Techne, responsible for testing antibodies for immunohistochemistry. We're doing it for a long time, and I think we gained some experience doing this stuff. And over the years, we learned a few tricks how to make the life of researchers easier and how to address few challenging things. And one of the big challenges in immunohistochemistry is the situation when researchers are stuck with having antibodies raised in the same host species. This is not unusual, and they cannot do, in this case, their immunohistochemistry because of the fact that secondary antibodies they will use will cross-react with all irrelevant ones, and they have a mess, overlaid images, overlaid colors, and they will not be able to identify the target of interest. Before we start, let's look at terminology briefly. As you all know, IHC stands for staining tissue sections, histone. Tissue sections. Quite often people use the term ICC, which stands for immunocytochemistry, and this stands for staining cells. So pretty much IHC and ICC referring to samples people use in their experiments. Quite often you also heard this term called immunofluorescence, which defines the detection technique rather than the sample type being under investigation. As you all know, that's the typical IHC workflow using the in immunofluorescence. First of all, cells and tissues have to be fixed using fixatives which will do cross-linking, such as formaldehyde or formalin, or using fixatives which will precipitate antigens, and these fixatives are ethanol and acetone, and there are many other like this. Of course, after fixation, fixed cells and tissues then incubated with primary antibodies. Antibodies may be monoclonal, polyclonal, and lately gaining popularity, recombinant antibodies. Then this phase is followed by incubating with secondary antibodies and detection reagents. And finally, a lucky moment, we can examine stained tissues under the microscope and submit our application for Nobel Prizes sometimes. What are the critical stages or events which will determine the IHC success? First of all, we need to preserve tissue antigens. Otherwise, we will not be able to detect them, no matter how good our primary antibodies are. We have also to preserve cell and tissue morphology, because we need to be as close, they need to be as close to their original state as possible. Of course, the critical things are quality of primary antibodies. And quality, by quality, I mean that the antibodies need to be of high affinity and high specificity. And the same applies to quality of secondary antibodies and detection reagents. Secondary antibodies have to be of high quality. They have to be purified against irrelevant targets, and they should interact only with your primary antibodies of interest. And quite often, when people deal with immunofluorescence, they think, you know, we're all obsessed with the intensity of fluorescent dyes, fluorophores, attached to secondary antibodies, forgetting that another huge component is the antibody itself. And I can give you a good example. If we are using secondary antibodies, which are, so to speak, lousy, they have low specificity, low affinity, and we conjugate them to brightest fluorophores, what are we going to have? We're going to have a mess, stained tissue with a lot of artifactual staining, a lot of non-specific background staining as well. That's why we at R&D Systems, we put a lot of effort into developing secondary antibodies of high affinity and high specificity. So that's, believe it or not, that's a lot of work. Sometimes it's even more complicated than to make new primary antibodies against novel targets. So why do we want to do multicolor fluorescence? Okay, first of all, this technique allows us to do simultaneous detection of multiple antigens. multiple. That's the key word. Second is that with fluorescent probes, we can get high image resolution and high contrast. And this is in comparison with techniques when we use enzymatic techniques like phosphatase peroxidase and diaminobenzidine. Comparing with these chromogenic bright field techniques, fluorescence has much stronger resolution and contrast. Now, we can detect, again, both overlapping and non-overlapping antigens. For example, this is hardly to do when people do multicolor staining using enzymatic chromogenic detection, because if antigens overlap, we have no means to pull out these colors or differentiate which color belongs to which antigen. Of course, fluorescence is a very convenient tool when we need quantitative image analysis. You know, there are platforms like high content screen platforms using fluorescence detection reagents when we don't only see through the cells and say yay or nay, it's a good signal or low signal, when we can measure simultaneous targets, different colors with regard to their intensity, the volume, the area occupied by the stain, their localization. And lastly, I would like to mention that immunofluorescence is an incredibly good tool for doing three-dimensional reconstruction of the distribution of antigens in cells and tissues. So with no other technique allowing to do this. And again, we're back to square one. What if we are stuck with the antibodies raised in the same host species? Now let's refresh our memory about the conventional protocol that we use in, let's say, for the sake of example, we're using three-color immunofluorescence detection here, and this is how we do it usually. We're lucky if we have 3 antibodies against 3 different tissue antigens, and all 3 antibodies raised in different species. Like here we show we have rabbit, mouse, and goat primary antibodies, and then we do our second round of staining using detection antibodies, secondary antibodies, which are raised against rabbit, mouse, and goat species, and these antibodies conjugate to fluorescent dyes, and now we got nice staining. There's no crosstalk between these antibodies. Goat antibody is not going to interact with mouse primary antibody, so we're not worrying about this. This is our classic conventional detection. Now, let's say we have only antibodies raised in the same host species, and to make this, to draw this awful scenario, I would like to put this cartoon. Let's say we have antibodies made in mouse. Primary antibodies are mouse, and we now raise them against different tissue targets, and we coming up with the antibodies, detection antibodies conjugated to different fluorophores, and now you see that all the secondary antibodies are going to interact with primary antibodies, and in the end we're going to see the image where all these colors overlay, and there's no ways for us to say or define determine which antigen has been detected. So what's the solution? And there are pretty much 2 solutions that can be used. One of them is to do a so-called direct detection technique. We can modify primary antibodies by conjugating them to fluorophores directly. This is a very nice setup when we conjugate them. It's one incubation step. We can just incubate cells in tissues with these conjugated primary antibodies, and then we're done. Take them and look on the microscope and get nice looking images. Another way of doing things would be to do indirect detection when primary antibodies are not conjugated to any fluorescent molecules. They are rather conjugated to small heptane molecules like deoxygenin or biotin. Okay, that's another approach. And here, let's talk about pros and cons of each technique and see how this can be used in practice. This is an example schematics of the direct detection if we conjugate our primary antibodies to fluorescent dyes, and that's exactly what we're seeing. We see nice separation of colors. These antibodies interact with their target molecules, and we have no worries about cross interactions, unless our primary antibodies are not so specific and they cross-react. That's inherent nature of the primary antibody. And this is an example of using direct multicolor IHC. You can see in HeLa cells we can detect vimentin, AIF, that's the mitochondrial biomarker, and LAMP, lysosomal biomarker, and you see there's a nice individual colors, and after merging them all, including DAPI counterstain, which counterstain nuclei, we got this intense image, but still we will be able to see and define the colors here and identify which color is localized to which compartment of the cell. That's easy. The only drawback with direct multicolor IHC that for unknown reasons, after conjugation of primary antibodies to fluorescent dyes, antibodies may get inactivated in terms of their affinity may go down. So we have to use high concentration of conjugated antibodies compared to unconjugated ones. In the worst case scenarios, what happens, we may have complete inactivation of primary antibodies. So that's what we have always to watch for. And always have to compare conjugated antibodies versus unconjugated to make sure that the quality affinity did not drop for primary antibodies after the conjugation. Now let's talk about the different technique that is indirect detection. And as you can see on these schematics here, this is a combination of the conventional immunohistochemical approach, immunofluorescence, and something that's not that very conventional. So that's how we do it. What we need to do to avoid cross-reactivity of secondary antibodies with the primary antibodies, irrelevant primary antibodies, we have to avoid using the antibodies, secondary antibodies, against the same primary antibody species. And here's how we do this stuff in this setup. First, we incubate tissue samples with primary antibodies like in conventional IHC, followed by incubation with a secondary antibody conjugated to fluorescent dye. That's the first step. It has to be done full cycle. After this is done, we now can incubate the same tissue section. Of course, I'm not talking about washes between the incubations, but that's, I guess it's understandable. So there are washes between the incubation steps. And then this is followed by incubating the same tissue sections with primary antibodies conjugated to biotin and the small heptane molecule called deoxygenin, abbreviated as DIG. This molecule is quite well known to molecular biologists who are doing in situ hybridization, because that's what they quite often use for the detection. This can be done also with this immunofluorescence, and now then we incubate with a mixture of these two antibodies, and then we incubate tissue sections with a mixture of these two detection reagents, I would call them. One is the streptavidin conjugated to fluorophore, and another is the antibody raised not against the IgG itself, but raised against the small molecule deoxygenin. Now, this antibody has no option to interact with the IgG, this one or this one or this one here. And that's how we do it. So 2 rounds, first cycle, go over again. We do this conventional, one antigen detection, followed by incubated with a mixture of these primary antibodies, followed by incubating with a mixture of these two detection reagents. One is the antibody, one is the streptavidin conjugated to fluorescent dye. Now, this is a nice cartoon, right? And I think the scholars look nice here, but how this is going to work in reality. And I'm going to show you a few examples when we use primary antibodies raised in different species. This is the example of using all mouse primary antibodies on a rat brain. And here we are detecting calbindin, synaptotagmin, and CART. And here for calbindin we use unmodified antibodies against calbindin, followed by incubated with anti-mouse Northern Lights 637 secondary antibodies. And then there was a mixture of incubating tissues with a mixture of the antibodies against synaptotagmin labeled with biotin and antibodies against CART labeled with deoxygenin. Okay, and then we used streptavidin conjugated Northern Lights 493, there's a green color here, and Northern Lights 557 anti-DIG conjugated antibodies, giving us red color here. As you can see, after merging all these colors into these 4-color image, yes, of course, DAPI uses a counter stain, cannot avoid using it. And then you see on the final image, resulting image, we still see separation of colors, and they there we can tell the we can define individual targets by just looking at the individual colors. Now, how about other species? Maybe it works only for mouse. It appears that this technique works also for goat primary antibodies, and that's another example when all antibodies are primary antibodies are raised in goat. Again, we are using unmodified antibody. This is the red cerebellum. You recognize it by the morphology. These are the Purkinje neurons, largest neurons in our body, and we are doing staining three main colors. We are using antibodies against E-cadherin, they unmodified, followed by plain anti-goat NL637 secondary antibodies, fluorescent antibodies, and then a mixture of the biotin-labeled anti-neprilysin antibody here, conjugated to biotin, and the antibodies against nephrin conjugated to digoxygenin, followed by, again, anti-teach antibodies conjugated to NL557, that's a red color, and here we got the green color conjugate, and now we got, when we merge all the images, we got this nice picture where we see still all the colors are visible, and we can tell the difference between different colors and identify them individually. Now, what's more interesting, that first of all, there is another species which are quite popular among researchers, and I'm talking about rabbit antibodies. Rabbit antibodies are popular because in academic labs, rabbits are the best source, best object, best host to make antibodies. We all coming from academic labs where we used to make our own antibodies, and rabbits were our species of choice. And interestingly enough, that rabbit antibodies appears, when it comes down to looking at phosphorylated proteins, it appears that rabbit antibodies are working the best when it comes down to detecting phosphorylated proteins. And that's why we are here using rabbit antibodies to see if our approach will work for detecting the phosphorylated proteins in tissues. This is an example of the red brain. This is a dorsal root ganglia where we see sensory neurons here. And again, we are using the same approach. We incubate first the tissue with unmodified anti-phospho-ERK1/2 antibodies here, followed by anti-rabbit NL637, that's a far red emission range, and then we do incubation with a mixture of antibodies against phospho-RET and phospho-RSK1/2. They are conjugated here to biotin, and this one is conjugated to digoxygenin, and followed by incubating with anti-DIG antibodies and streptavidin conjugated with the fluorescent dye NL493. And again, as you can see, after merging all the images, we still see individual colors and can define them clearly on this image. So it appears that this approach works nicely for different antibodies raised in different species, and I'm sure that this approach will also work with antibodies raised in guinea pigs or chicken, name it. So this is just the way to go, and the good way to avoid getting cross-reactivity between secondary antibodies and primary antibodies. Of course, when we do this technique, we have to make sure that after conjugation, our primary antibodies do not lose their affinity. It's okay for them to lose some affinity, which means that the concentration of our conjugated primary antibodies may be higher compared to unconjugated ones. This is normal, and this is what we do regularly here, these tricks with our antibodies, and we see it, and there's nothing unusual about it. We know that after conjugation it may happen. Now, this all techniques are really nice, but again, as you can see, we have to do the first round of primary, secondary, and second round of primary antibodies conjugated to heptans like biotin and deoxygenin. How nice would it be if we can conjugate all primary antibodies to different heptans, okay, and then use secondary antibodies against each individual heptan, and this antibodies conjugate to fluorescent dyes, then our life will be much, much easier. We will do incubation first with the mixture of primary antibodies conjugated to all different heptans, followed by incubation with anti-heptan secondary antibodies. This is too good to be an easy solution, because the problem is to find these heptans, what can be used as heptans, and if they are good enough for detecting with the antibody that can be raised against them. But we know, as far as we know, there are different companies working on this approach, trying to identify them. And one of our collaborators recently just, they developing this technology, this heptans, the small company, they sent me this image showing how this can be done. And this is pretty much, like I said, you incubate all your sample with primary antibodies and then with the anti-heptan secondaries, and that's what you can get. What you get pretty much here is the same result what you get with the approach I described before, but it just adds some convenience. Okay. One of the drawbacks maybe of this technology is that you can, for example, if you want to biotinylate your antibodies, you can do it by yourself. You can do it on the bench. You can buy conjugation kits. There are many companies offering them. And you can also conjugate to biotin. You can conjugate to deoxygenin. That's easy. When it comes down to things like this heptans developed by other companies, it may happen that it's not easy to do this conjugation, because quite often they don't tell you what this is, and they tell you, okay, well, this is the antibodies that see them. So these are the pros and cons of using different approaches. But in the end, the readout is the same. We got to get separation of colors and identify different targets in the tissue. Like here you see progesterone receptor, estrogen, Ki67, and HER2. So now let's wrap it up and leave time for questions. So in the summary, what I would like to say, so multicolor IHC with antibodies from the same host species is easy to do. Anyone can do this. Just little practicing and this can be done. Multicolor staining can be done with direct or indirect techniques, and if you stretch our imagination, we can even combine them. We can use one primary antibody directly conjugated to fluorescent dye, and then other antibodies can be conjugated to different heptans. So that's just a way of doing these things. And I have to emphasize that specificity and affinity of primary antibodies is of critical importance. And that's what we spend a lot of time on developing such antibodies which have this high quality. So after conjugation, they don't lose their affinity, or if they lose it, it's still doable to do multicolor staining. And of course, a control that has to be done is to, after modification of primary antibodies, they need to be tested first in single color IHC staining and compared against the unconjugated antibodies. Well, let me stop here, and I'm ready to take questions if you have them. Thank you.
Thank you very much, Alex. We do have some questions that have been typed up there already. So we have one from Susan Ghazi. I'm just going to read this out because there are actually quite a lot of questions. Do you have any tips for using primary antibody raised in the same species as the tissue sample? So, for example, using a mouse primary on a mouse tissue.
It's a very good question. I was going to mention this, but because of the lack of time, yes, indeed. This approach, this trick what we do here, can be also used with mouse-on-mouse detection. And I can tell you what's happening. When it comes down to CNS, it's not a big problem. We can use mouse brain and we can use mouse primary antibodies because, as we all know from histology classes, the brain is above blood-brain barrier. So there are no IgGs floating around. So there's no cross-reactivity of secondary antibodies with IgGs in brain. Okay, unless there's like cases when there is some damage to the brain. Okay, that's no problem. But let's say we're trying to stain lymphatic tissue, spleen, lymph node, tonsil tissue. That's then it's a big problem, because our secondary antibodies, no matter how good we do our blocking of our mouse IgGs in this tissue, it's impossible to block all of it. There are companies selling so-called mouse-on-mouse blocking kits. We tried them many times with mixed success, but it's impossible to block all the staining. What we do now, we can conjugate our primary antibodies, for example, to deoxygenin. And the beauty of deoxygenin is that this is a plant protein. It's not found in animal tissues. So if you use anti-DIG antibodies, there's nothing to detect. There's nothing to cross-react with. So yes, the answer is yes, you can use it when you use your primary antibodies on the same, and when you have to use secondary antibodies on the same host, on the same species where these antibodies were raised in. Okay. Other questions, please.
Thank you, Alex. Another question here was to do with the adding of the antibodies. So do we mix together the primary antibodies in one cocktail, and also do you incubate the secondary antibodies together?
We do them. We mix them together in. I assume the question is about when we do the staining with the antibodies in the same host species. We do combine only primary antibodies which were modified. We do not combine unmodified and modified. Modified, I mean conjugated to haptens primary antibodies. That's why we do them in separate steps. Okay, but we can combine them together and we can use them as a mixture. So that's the answer is yes.
Okay, that's great. Thank you. Somebody's asked here, can we get more details about the resources and methods used to conjugate the antibodies?
That's a good question. We do not make any conjugation kits here at this point, and you may try to find a conjugation chemicals from other companies. Thermo Fisher, I'm sure, has a lot of them, and other chemical companies. So they are available off the shelf and can be found easily if you just Google it.
Thank you. Does the conjugation to biotin or deoxygenin decrease the affinity of the primary antibody in a similar way to fluorescence conjugation?
It's yes and no. It's yes and no, and to be precise in saying what's the extent of the inhibition of the affinity, we have to do side-by-side comparison, conjugating primary antibody to the fluorescent dye and conjugate to deoxygenin, and we have never done these experiments, but I would say we have to assume that after conjugation the affinity may drop. Affinity will drop because we don't know, first of all, we don't know where the hapten goes on the primary antibody. It may bind to FC part, which is fine, that's what we expect, we want to happen, but it may also bind to FAB part, the part of the antibody that is binding to the antigen. And we may not be able to control how many molecules, where these haptens go on the primary antibodies. I would say I would be cautious when do conjugation. Yes, we sacrifice some affinity, but this can be leveraged by increasing the concentration of our primary antibodies, as well as increasing the concentration of our secondary detection reagents.
Another question here. I'm told that conjugation with haptens can interfere with primary antibody binding to the antigen. Have you observed this, and is there any way to get around it?
Well, we have observed it, and the way to go around this, like I mentioned, said in my previous answer, is to increase the concentration of primary antibodies and increase the concentration of secondary antibodies. So we have to sacrifice. Conjugation has a negative effect on affinity of the antibodies, we know this, but if there is no other better way of doing stuff, we have to accept it.
Okay. The question here about using cultured cells. Are there any tips for using direct technique on human primary cultured cells?
Oh, the same technique can be applied to human primary cultured cells. That's exactly the same way to do it. The only thing you need to find out is which fixation is going to be the best for your sample. Is it going to be formaldehyde, cross-linking reagent, which cross-links proteins, or precipitating fixatives like acetone, alcohol, which will precipitate your targets? We found that on cells sometimes the precipitating fixatives give better results. That's so you need to test this as well, so to see which one works the best. But yes, absolutely, you can do it. You can fix your cells. You can add your primary antibodies to the cells, like we use quite often the so-called chamber slides when we pour antibodies right there, and then can remove the chamber at the end of the experiment. Yes, absolutely, it's applicable to any sample.
We've got a couple of questions here about antigen retrieval, about top tips for successful antigen retrieval. And someone says that they've had problems that sometimes the antigen retrieval method can actually damage the tissue or make it come off the slide. And so labeling becomes quite difficult. So have you got any tips or suggestions when you're trying to use antigen retrieval method?
Yes, we do. Antigen retrieval is done on all our antibodies when it comes down to do their testing validation on human tissue sections. And this is a very good point. We also see it in our lab as well. And the options are here to find the better slides. I mean, the slides which have better adhesion for the tissue sections. Okay, and that's just only trial and error, going through different vendors and see which slides from which vendor works the best. Another way to do retrieval would be you can reduce the temperature, because it goes into either vegetable steamer or the special anti-clocking, declocking device, so-called, where temperature is very high. It's like it's almost 98 degrees C. It's almost close to boiling. You may want to use it and try to use lower temperatures, go to 70 degrees and see how this will work. These are the only suggestions. I can tell you that paraffin-embedded tissue sections are less vulnerable to antigen retrieval procedure versus cryostat sections. Cryostat sections or frozen tissue sections are really sensitive, and you're correct. There's a physical damage to the tissue. Yes, that's right. But only trying to find better slides that will get the tissue will stick better to the slides. That's the only way to do it.
And I suppose the related question is, because we're looking at several antibodies at the same time, each antigen might not necessarily be retrieved equally. So if every antigen is needs a specific antigen retrieval system, can you actually do more than one antigen retrieval on the same tissue section, or is that counterproductive?
Well, it's when it comes down to experimental biology, nothing is impossible, and we have never done this. I would say we don't do it regularly. We've done it a couple times, but yes, it's possible to combine different techniques, different retrieval techniques, and different, I mean, that you can use, because what we do with retrieval, the temperature conditions are pretty much the same. We're changing the solutions. They have different pH. We can go with a low pH, pH 6. We go with high pH, pH 9 or 10, and why not? It's quite interesting to try. But and then what's important here is to see which retrieval technique should be done first or gives better results. You go with a high pH one, or you go with a low pH one. Easy to do, easy to try on the bench. But before diving into multicolor staining, of course we have to do single color detection to make sure that antibodies are detecting these antigens on the tissues, how we process them.
We have a question about autofluorescence as well. So is there a way of reducing background autofluorescence on your tissues?
It depends it depends on what autofluorescence is, where it's coming from. There are endogenous substances which autofluoresce, and it's really hard to quench their autofluorescence. And good example would be so-called lipofuscin. That's the agent pigment stored in lysosomes, which fluoresces under different filters and gives us red, green, blue, all sort of fluorescence. And even if sometimes when you look at the stained tissue with the antibodies conjugate to fluorescent dyes, you may see of your specific staining. This non-specific staining is not going to fade. Okay, if you want to keep your tissue for a long time, it's not going to be gone. So there are several solutions you can try. There are several reagents that can be used. One of the reagents is to use a compound called Sudan black. And there was a paper published many years ago on how to use it, I believe, in Immunohistochemistry and cytochemistry. Just Google Sudan black and autofluorescence. Sudan black binds to lipids in the tissue, and it quenches the autofluorescence. But when you use Sudan black, you need to be sure to make sure that after staining the tissue, you don't dip it into alcohols and xylenes, because sometimes when we do permanent mounting using xylene-based mounting media, we have to walk slides through alcohols and xylenes. Alcohols and xylenes will dissolve Sudan black. That's a drawback. So you need to use with Sudan black, you have to use the aqueous mounting media, which is not a big deal. So that's a solution. Okay. Another solution of quenching endogenous autofluorescence, people sometimes use copper sulfate. Also, you can Google it and find out on the web, online, how to do these different solutions. But yes, this is a big problem. And the only way you do it is to use these treatments, or you need to find different labels. Instead of fluorescence, you may want to go to chromogenic. So that's a big issue, and unfortunately, it's not solved yet. How to block autofluorescence in tissues.
Okay, we're experiencing a little bit of sound interference, so I think what I'm going to do is actually stop the event here. Any questions which have not been answered, we will receive your answer by email, so don't worry about that. I'm sorry for those who didn't get their questions online. Thank you, Alex, and there will be a recording made available so everybody can receive a link to the recording. Thank you very much.